Welcome all to the today's session. So today we continue with biochemistry module and we will have the recap of carbohydrate metabolism. This topic is very important from the examination point of view, and at least 3 SBAs you will expect from this carbohydrate metabolism. So what is the definition of carbohydrates? These are organic molecules composed of carbon, hydrogen and oxygen. So the general formula is CH2O and outside the bracket it's N. They are the major source of energy for the human body. So what are the types of carbohydrates? See, these carbohydrates are of three types, that is monosaccharides, disaccharides and the polysaccharides. Here they can ask you the questions, which are the monosaccharides, which are the disaccharides, etc. They can ask the examples, okay, and they can give the permutations and combination. Following are the monosaccharides out of these. Okay, now the monosaccharides are the simplest form of carbohydrates and the examples are the glucose, fructose and galactose. Disaccharides are the two monosaccharide units. They will form the disaccharides and the examples are the sucrose, lactose and maltose. And polysaccharides are the long chain monosaccharides. These are the complex carbohydrates. These are the starch, glycogen and cellulose. Okay, so when they are asking the question, they might ask you the example of disaccharides and then they will give you five options, each having three. But only one option will be correct, that is sucrose, lactose and maltose, but they may have the combination of sucrose, glucose, starch, etc., etc. So you should know the correct group, which are the examples of disaccharides, which are the examples of monosaccharides, etc. Okay, so concentrate this carefully. Then coming to the complex carbohydrates, these are polysaccharides. So polysaccharides are the large molecules made of multiple monosaccharide unit. So these are the large molecules made up of multiple monosaccharide units, and the function is they act as an energy store. For example, in plant, there is a starch. The glycogen is present in the animals. These are the energy storage of from the carbohydrates. Then they may be component of structures. That is, these are the structural components. Like in plants, it is the cellulose. Then in insects, it is the chitin, which is the structural component. Then how about the digestion of the carbohydrates? The carbohydrate digestion converts complex carbohydrates into monosaccharides, which can be absorbed. So the digestion will involve the conversion of the complex carbohydrates into monosaccharides. And these are the monosaccharides which can be absorbed. Okay. So carbohydrate digestion occurs in three main sites. So three sites are the mouth, the small intestine, and the brush borders of the small intestine. So in stomach, there is hardly any digestion. There is a very minimal digestion. So main are the mouth, the small intestine, and the brush borders of the small intestine. Okay. So mouth, there are salivary amylase, and also there is ptyalin. Then action, it begins the starch digestion. So in mouth, what happens? There is a digestion of the starch, and the products which are formed are the maltose and the dextrins. Then in the stomach, there is a hydrochloric acid. In this acidic pH, there is no action of the salivary amylases. So there is stops salivary amylase activity because of this acidic pH, and there is a minimal digestion of the carbohydrates in the stomach. Then next is the small intestine, where there are pancreatic amylases. There is a they will continue the starch digestion and you will get the maltose. Okay. Then you have the brush border of the intestine. Then in the brush border of the intestine, what is present? These are the disaccharides which are present. They will convert disaccharides into monosaccharides. Okay. And you will get the monosaccharide like glucose, fructose and galactose. These are monosaccharides. So what are the brush border enzymes? There is a maltase, which will convert maltose into two molecules of glucose. There is a sucrase or isomaltase that will convert sucrose and isomaltose into glucose and the fructose. Then there is a lactase that will convert lactose into glucose plus galactose. Okay, so always glucose is there. Along with that, there will be either fructose or galactose or glucose plus glucose. Okay, so maltase, sucrase, and lactase. Again, remember this. This can be asked in the SBA. Okay, so final end products are the monosaccharides. These are the glucose, fructose, and galactose. So these monosaccharides are absorbed across the intestinal epithelium into the bloodstream. So finally, the absorption will occur when ultimately it comes down to the monosaccharide level, that is glucose, fructose, and galactose. Now, how does the absorption of the carbohydrate takes place? It occurs mainly in the duodenum and jejunum. This can also be asked as a SBA. Where does the carbohydrate absorption? It is a duodenum and the jejunum. Now concentrate on the transporters. Okay. See, now there is a digestion. We are now at the level of the absorption. So glucose is there. Now there is a sodium dependent active transport, and the transporter which is used is a SGLT1. For galactose, there is a sodium dependent active transport, and the transporter which is used is SGLT1 again. For fructose, it is a facilitated diffusion, and the transporter is a GLUT5. And for pentoses, it is just a simple diffusion, which is passive in nature. Okay, so once they are converted into monosaccharides, then they are transported through some mechanism. Okay, last time we have discussed about the simple diffusion, facilitated diffusion, transport, etc., etc. Okay, what are the different methods of transport? Remember, for glucose and galactose, you have sodium dependent transport, and the transporter is SGLT1. Okay, for glucose and galactose, you need a transport. For fructose, there is a free entry. It is a facilitated diffusion. Okay, but you require a transport, that is GLUT5. But it is a free entry by facilitated diffusion. And pentoses, it is a easiest entry by simple diffusion. Okay, it is just a passive transport from high gradient to the low gradient for pentoses. For glucose galactose, it is SGLT1. Okay, for fructose, it is a GLUT5. So glucose and galactose, you have SGLT1 as a transport. Okay, transporter, and mechanism is a sodium dependent, that is secondary active transport. So remember, Gg goes with the sodium. Okay, glucose and galactose goes with the sodium. Okay, fructose has got a AZ entry. What does it use? It use GLUT5. Mechanism is a facilitated diffusion. So fructose has got a free entry via facilitated diffusion. Then you have pentoses is the simplest entry. Mechanism is a simple diffusion. No transporter is needed. Okay. Now, from intestine, how these sugars will go into the blood. Now, what we have seen from the lumen, it goes into the intestinal enterocytes. Okay. Now, from intestine, how it will go into the blood. So, for all sugars will exit via GLUT2. So, there is a common exit gate. So, for entry, there were different gates for the glucose, galactose, fructose, etc. But for exit, there is only one gate, that is GLUT2. Okay. So for fructose, we have seen entry gate was GLUT5. Okay. For galactose, it was a SGLT1. But for exit, for everyone, it is a GLUT2. That is the common exit gate. So just remember, all sugars will exit via GLUT2. Okay. So after entering into the intestinal cell, sugars exit the blood via GLUT2. Now different entry and same exit is there, that is GLUT2. Okay. Now glucose transporters, these also you can remember, there are different types of transporters which are present in the different tissues. Okay. See, GLUT1 is present in the brain and RBC. So function is the basal glucose uptake. Remember, GLUT1 is the brain and RBC, because brain and RBC require glucose for their metabolism continuously. Okay, so the priority is GLUT1. Okay, then second is actually it acts as a glucose sensor. GLUT2 is present in the liver and pancreatic beta cells. Here they are acting as a glucose sensor. They will decide the blood glucose level, and accordingly they will behave. So it is a second comes a GLUT2. Third is again neurons. GLUT3, it has got a high affinity for the glucose uptake. Then next is a GLUT4, that is muscle and adipose tissue. And remember, it is insulin dependent uptake. For GLUT4, it is always insulin dependent uptake in the muscles and adipose tissue. Then you have SGLT1 and SGLT2. SGLT1 we have just seen, it is present in the intestine. It is responsible for absorption of the glucose and galactose, and SGLT2 is present in the kidneys. It is responsible for the reabsorption of the glucose in the kidneys. Okay, so GLUT1, GLUT2, GLUT3, GLUT5, SGLT1 and SGLT2, you should be able to remember. GLUT1 in the brain and RBCs, GLUT2 liver and pancreas, where it is these are acting as a sensors. GLUT3 is neurons, which has got a high affinity for glucose uptake. GLUT4 is insulin dependent and it is present in the muscle and adipose tissue, while SGLT1 we have seen in the intestine and SGLT2 is present in the kidney. Okay, so you will remember this. GLUT1, brain and RBC, it is just the baseline supply to the brain via GLUT1 function. There is a constant glucose supply. Even if you are starving, brain and RBCs must survive. Okay, so it is through GLUT1. Then GLUT2, it is a sensor and regulator. So liver and pancreatic beta cells have GLUT2. Function is a glucose sensing. So GLUT2 checks how much is the blood glucose in the blood, and accordingly it will behave. In pancreas, it will trigger the insulin release, and in liver, it takes the glucose after the meals. Okay. In GLUT3 is in the neurons. Okay. Function, it has got a high affinity uptake, and neurons grab the glucose even when the levels are low. That is how you remember. For GLUT4, it is a insulin dependent, very important. It is present in the muscles and fat, and function, it will respond to the insulin because it is a insulin dependent. GLUT4 is a insulin dependent. So after the food, insulin opens GLUT4 doors. So that is the basis for diabetes and insulin resistance. Now SGLT1 is present in the intestinal absorption, and the function is a glucose and galactose absorption with sodium. Okay, this we have already discussed at the time of absorption of the carbohydrates. Then SGLT2 is present in the kidney. Here it saves the glucose. Okay, it is present in the kidney proximal convoluted tubules. Function is a reabsorption of the glucose, and kidney doesn't want to waste the glucose. Okay, so it is through the SGLT2, and if the patient is taking SGLT2 inhibitors, that will cause the glycosuria. Okay, now from the examination point of view, remember, for brain you have GLUT1 and 2, GLUT1 and 3 that needs constant supply. Then GLUT2 is present in the pancreas and the liver. Okay, so in pancreas, it will check how much is the glucose, that is how much money is there. Liver will store the extra money. Then muscles and fat will spend the money when there is a insulin. Okay, so it will act on the instructions of the insulin. So in the intestine, it will absorb the money, and in kidney, it is SGLT2 is present, it will prevent the money loss. Okay, so remember, in this particular way, you will be able to remember all the transporters. Okay, so GLUT1 is a basal, GLUT2 is a sensor, that is liver and pancreas. Then GLUT3 is again a brain priority, and GLUT4 is a insulin dependent muscle and adipose tissue. Okay, now coming to the clinical disorders. You have a lactose intolerance. This is mainly because of the lactase deficiency. It may be congenital or primary adult type or secondary intestinal disease. Pathophysiology is that undigested lactose will be fermented by the bacteria, and you will get short chain fatty acids plus gas. Okay. So the patient will have osmotic diarrhea, abdominal pain, flatulence, bloating, and diagnosis is by hydrogen breath test. There will be acidic stool. Then next is a deficiency of SGLT1 will lead to the glucose galactose malabsorption. We know that SGLT1 transport is required for glucose as well as galactose. So glucose galactose malabsorption will be there. Features will be severe diarrhea in infancy, dehydration, and it improves when the glucose and galactose are removed from the diet. Okay. Now what is the fate of absorbed glucose? Now we have seen the absorption of the glucose. What will happen to this glucose after the absorption? So it can undergo glycolysis, that is for energy production. Then it may undergo glycogenesis. So glucose will be converted into the storage form, that is glycogen. Then glycogenolysis. So whatever is the storage form, glycogen, that will be broken down. That is glycogenolysis. Then gluconeogenesis, that is the formation of the glucose. But this is not from the carbohydrate substrate, it is from the non-carbohydrate substrate the glucose is formed, that is called as gluconeogenesis. Then there is a HMP shunt, which is responsible for NADPH production, and the Krebs cycle is important source for the ATP. Okay. Now first we will see the glycolysis. So glycolysis is a metabolic pathway in which glucose is converted into pyruvate in the aerobic condition or the lactate in the anaerobic condition with production of ATPs. Okay. So this word glycolysis is derived from the Greek word glykes, means sweet, and lysis is a splitting. Okay, what are the sites of glycolysis? So it is the cellular location is in the cytoplasm. This is the SBA which is asked, where does this glycolysis happen? It happens in the cytoplasm. Then oxygen requirement, it occurs in the both aerobic as well as anaerobic condition, and it occurs in all cells of the body. Okay, so what is the significance of the glycolysis? It is a universal pathway occurring in all cells. Then only source of ATP in the RBCs. RBCs does not have mitochondria. So only source of ATP in the RBC is by glycolysis. It is the major source of energy in sternus muscle activity that is under anaerobic condition. It acts as a first stage of glucose oxidation before TCA cycle. So before it goes into the TCA pyruvate, glucose will be converted into pyruvate, and that will further enter into the TCA cycle. So before the TCA cycle, the first stage is of glucose oxidation is glycolysis. So it provides carbon skeleton for non-essential amino acids as well as glycerol, a part of triglycerides. So many reactions are irreversible, allowing the gluconeogenesis. So in glycolysis, there are many steps which are reversible and they will allow the gluconeogenesis. Okay. Now, what happens?
This is a reminder.
Alexa, stop.
Near the
So cellular utilization of the glucose. So after absorption, glucose is transported to the tissues and it undergoes cellular respiration to produce the energy. So what is the goal of cellular respiration? It is the production of ATP, that is adenosine triphosphate. Now why this ATP is required? ATP is required for the muscle contraction, active transport, biosynthesis and cellular functions. For all these functions, you need an ATP. Okay. So what are the major steps of cellular respiration? First is a glycolysis. What is the location? It happens in the cytoplasm. This is a SBA. They will always ask you the location of the various processes which are happening. Glycolysis, it happens in the cytoplasm and the glucose will be converted into pyruvate. Okay. Glucose is a 6-carbon compound. Pyruvate is a 3-carbon compound. So from one glucose, you get 2 pyruvate. Okay. So conversion to acetyl-CoA happens in the mitochondria. So pyruvate will be converted into acetyl-CoA. It undergoes citric acid cycle, also called as TCA cycle, in the mitochondria. There is the oxidation of the acetyl-CoA and further through the electron transport chain, which is located at the inner mitochondrial membrane. So ETC, that is electron transport chain, is located on the inner mitochondrial membrane, and that is responsible for the ATP production. Okay, that is how the ATP is formed. We will discuss about the glycolysis. So it is the metabolic pathway in which glucose is converted to pyruvate or the pyruvic acid with the production of ATP. Now they ask you in the SBA, what is the location of this particular process? Where exactly this happen? They can ask this for the Krebs cycle. They can ask for glycolysis or various. So always remember the location. Whenever the location will come, you have to remember from SBA point of view. So the glycolysis location is cytoplasm. Okay. Now you should remember that this one glucose, which is a 6-carbon compound, when it undergoes glycolysis, it will give you 2 pyruvate molecules. So pyruvate has got 3 carbon each. Okay, so from one 6-carbon compound, that is glucose, you will get 2 pyruvate molecules which have 3 carbon atoms. Okay, now what does this glycolysis produce? It will produce 2 ATP and 1 NADH. Okay, so this is the thing which we have learned in our first MBBS. This flowchart we have learned. But there is no need to remember each and everything. All the steps are not required. Just remember there are total 10 steps, out of which these are the enzymes which are there on the left hand side. These which are written in red, they are the rate limiting enzymes, and these are the steps which are irreversible steps. So rest of the steps are reversible steps, so they can work in either direction. However, these steps are irreversible steps. So first is a hexokinase. The next enzyme is the phosphofructokinase, and this is the 9th step, that is pyruvate kinase. So step 1, 3 and 9 are irreversible steps. Okay, now you can see in the first step and in the third step, one ATP is getting utilized. So here one ATP is getting utilized and ADP is getting formed. Here also from ATP you get ADP. That means there are two ATPs which are utilized. Now once this glycolysis is started, it will break this 6 carbon compound into two molecules of 3 carbons. Okay, somewhere in the middle it will have two molecules, and these two molecules will further have two same sequence of the events which are happening. Okay, so now concentrate at step 5. At step 5, what is happening? NAD is getting converted into NADH. So there it is, one NADH is getting generated. Okay, so step 5 is coupled with the step 10, where pyruvate is getting converted into the lactic, and the NADH which is formed with the in the step 5 is again utilized to get back the NAD. Okay, so this gets nullified. Okay, this happens under the anaerobic conditions. Pyruvate is getting converted into lactate under the anaerobic condition. Okay, while under the aerobic condition, this can enter into the Krebs cycle, okay, or the TCA cycle. Now see the step 6 and step 9. Okay, step 6, one ATP is getting formed and one ATP is getting formed here. That means two ATPs are getting formed from one molecule. So, but there are two molecules, because this 6 carbon compound will get split into two somewhere in the middle. So it is getting repeated. So total 2 into 2, that is 4 ATPs getting formed. NADH also, 2 NADH will get formed. However, under the anaerobic glycolysis, it will get neutralized. Okay, so there will be no net NADH production under the anaerobic glycolysis. Only total 2 ATP in one and 2 ATP in the other molecule. So total 4 ATPs will be getting formed. Okay, but we have utilized the ATP in the step 1 and step 3. That means total gain will be only 2. Okay, so what is the fate of the pyruvate? So ultimately in this glycolysis, glucose will be converted into pyruvate. This pyruvate under the anaerobic conditions with the help of lactate dehydrogenase will get converted into lactate. So that is one fate. This is under anaerobic, and if there is oxygen which is available, this pyruvate will get converted into acetyl CoA. This acetyl CoA will enter into the citric acid cycle, also called as Krebs cycle or the TCA cycle. And when the oxygen is absent at that time, as I had discussed, it will get converted into the lactate under the lactate dehydrogenase enzyme. Okay, so this anaerobic glycolysis happens in the muscle which is exercising and in the RBCs. Okay, so let us talk about the energy yield. Before we go ahead with the energy yield, let us talk about the enzymes which are regulating the these glycolysis. Okay, so there are enzymes like hexokinase, glucokinase, pyruvate kinase, and phosphofructokinase. This phosphofructokinase is the main rate limiting enzyme. This is also important from the examination point of view. What is the rate limiting enzyme for the glycolysis? Okay, now there are certain activating factors and there are certain inhibiting factors. So if you see these enzymes of the glycolysis, that is hexokinase, glucokinase, pyruvate kinase, phosphorus, they are activated by the insulin, then fructose 1,6 biphosphate, okay, cyclic AMP. These are the activating factors, while inhibiting factors are the glucagon mainly and ATP, cyclic AMP, etc. Okay, so these are the inhibiting factors. Okay, now let us talk about the energy yield. So this flow chart step 1, concentrate here, step 1 and step 3, one ATP is getting utilized, one ATP is getting utilized. Now step 6 and step 9, one ATP is getting formed, one ATP is getting formed. However, there are two molecules. So two ATPs here and two ATPs in step 9. So, okay, two ATPs in step 6 and two ATPs into step 9. So total 4 ATPs are getting formed. Out of 4, 1 and 3 is getting utilized, 2. Okay, so 4 minus 2 is 2. That is the total net gain under the anaerobic condition. Okay. Okay, because NADH will not give you ATP because this NADH step 5 is coupled with step 10. Okay. Now what happens under the aerobic condition? So you should also know the energy yield. These are the SBAs which are asked. Okay. What is the energy yield in the anaerobic glycolysis? It is total 2. Okay. Now energy yield number of ATPs per molecule of glucose in the glycolytic pathway under aerobic conditions. That is the oxygen is available. The step 1 and 3, they will utilize 1 each. Okay. Now in step 5, one NADH is produced. Okay, one NADH is produced, that we know. Then step 6 and 9, 1 ATP is getting formed, but there are two molecules. That is why here you have 2 and 2. Okay, so these are the total 4 ATPs, and these are the 2 ATPs which are getting utilized. Then one NADH is equivalent to 2.5 ATPs. One NADH is equivalent to 2.5 ATPs. So that will give you 5 ATPs. So this 5 plus this 4 is total 9. 2 are getting utilized. That means 7. Okay. So under aerobic condition, glycolysis will give you 7 ATPs. Anaerobic condition, 2 ATPs. Aerobic conditions, 7 ATPs. Is that right? Is it clear?
Yes, sir.
Yeah. So I will just read out the same thing what I have explained. During anaerobic, that is the oxygen deficient condition, when one molecule of glucose is converted into 2 molecules of lactate, there is a net yield of 2 molecules of ATP. Okay, SBA question. Then 4 molecules of ATP are synthesized by 2 substrate level phosphorylation, that is step 6 and step 9, but 2 molecules of ATP are used in the step 1 and step 3, and hence the net is only 2 ATPs. Okay, this we have seen under the anaerobic condition. The whole reaction is summarized as glucose plus 2 pi plus 2 ADP gives 2 lactate plus 2 ATPs. Okay, but the oxygen is in plenty. The 2 NADH molecule generated in the glyceraldehyde 3-phosphate dehydrogenase reaction, that is step 5, can enter into the mitochondrial electron transport chain, and that will undergo complete oxidation. Okay, so as I have already mentioned, under the aerobic condition, what can happen? These two molecules can through the electron transport chain will give the further ATP production. So each NADH provides 2.5 ATPs. This reaction generates 2.5 into 2. That is why you will get 5 ATPs under the aerobic condition. Thus, when the oxygen is available, the net gain of energy from the glycolysis pathway is 7 ATPs. Hence, the ATP yield from glycolysis is different in aerobic condition and in anaerobic condition. In aerobic conditions, pyruvate is converted to acetyl-CoA, which enters the TCA cycle for complete oxidation. Complete oxidation of the glucose through the glycolysis plus citric acid cycle will yield a net 32 ATPs. Okay, so only glycolysis under aerobic condition will give you 7, but the complete oxidation, glycolysis plus TCA cycle will give you 32 ATP. Okay, so now how these 32, that also we will see. Okay, previously calculations were made assuming that NADH gives 3 ATPs and FADH gives 2 ATPs. This will amount to net generation of 38 ATPs per glucose molecule, but recent experiments show that these old values are wrong and the net generation is only 32 ATPs. So previously they used to say that complete oxidation of one glucose will give 38, because the calculation were dependent saying that one NADH will give you 3 ATPs. But as per the new calculation, one NADH is equal to 2.5 ATPs, and one FADH was considered as equal to 2 ATPs, but nowadays they consider one FADH is equal to 1.5 ATPs. Okay. So let us see this calculation. So glucose is giving 2 lactate. So ATP produced are 4. Net ATP are used are 2 ATPs. So net gain is 2 ATP under anaerobic condition when the oxygen is not there. So reaction becomes glucose plus 2 ADP plus 2 PI will give 2 lactase plus 2 ATP. This is under anaerobic condition. Now let us talk about aerobic condition. That is when the oxygen is available, you get 2 NADH formed in the glycolysis. So each NADH is equal to 2.5 ATP. So total NADH will give you 5 ATP plus 2 ATPs, that is the substrate level. So net glycolysis yield under the oxygen when the oxygen is available is 7 ATP. Now this is further pyruvate is getting converted into acetyl CoA. Acetyl CoA is getting involved into the TCA cycle, and that complete oxidation will give you total 32 ATPs. Okay. So the old value NADH was equal to 3. So there were 38 ATPs, but new accepted value of NADH is 2.5 ATPs. So total is 32 ATP. So this is also asked in the SBA. Okay. So you may have odd answer also sometimes. So if 32 is not there, then you can select 38 also. But look whether you are dealing with the only glycolysis or glycolysis plus TCA, and whether you are dealing with aerobic conditions or anaerobic conditions, and then you select the answer appropriately. So under anaerobic conditions 2 ATP, aerobic conditions 7 ATP from the glycolysis only, but the complete oxidation will produce 32 ATPs. Okay. Now we have certain questions. Dr. Sansi, can you read this question and attempt the answer?
Yes, sir. A carbohydrate that cannot be hydrolyzed further is called disaccharide polymer glycoprotein. Option E, sir.
Carbohydrate that cannot be hydrolyzed further is called as?
Option C, monosaccharide.
Correct, correct. This is the in the beginning only we have discussed. So monosaccharides.
Yes,
Monosaccharides are the simplest unit, and examples are the glucose, fructose, and galactose that cannot
correct.
be broken down further. Okay. Next
Which
question,
bond
doctor.
links to monosaccharide in a carbohydrate? Peptide bond, glycosidic bond, hydrogen bond, ester bond. Option C, sir, hydrogen bond.
Is it hydrogen bond, two monosaccharides carbohydrate?
Option B, glycosidic bond.
It is option B, glycosylic bond. Okay, that joins two sugar molecules. Okay, that for example, maltose, glucose plus glucose will form maltose. Okay. Next question, doctor.
Which enzyme initiates carbohydrate digestion? Pancreatic amylase, salivary amylase. Option B, sir, salivary amylase. Starts in the mouth.
Which enzyme initiates carbohydrate digestion? It is salivary amylase. It starts in the mouth. Digestion starts in the mouth,
Yes.
and it breaks the starch, and it will convert it into maltose. Next.
Where does most carbohydrate absorption occurs? Option C, duodenum and jejunum.
Correct, correct. So carbohydrates are getting absorbed into the duodenum and jejunum. Major absorption is happening here. Next.
Glucose enters intestinal epithelial cells via SGLT1. Option C.
SGLT1. Okay, we have seen that for transport of glucose, it is different, then galactose is different, fructose is different. Okay,
Yes.
so SGLT1 is the gate which is used for glucose transport. It is a sodium dependent transport, and also it transports galactose. So for glucose and galactose, you have SGLT1.
Fructose is absorbed via facilitated diffusion. GLUT5. Option D.
GLUT5, correct. So you have for fructose, you have GLUT5 and facilitated diffusion. Okay, no sodium is required. Next.
After absorption, glucose exits intestinal cells via GLUT1 and SGLT1.
Exit from the cells. There is only one gate. Entry gates are different, but exit gate is only one. Which is that gate?
GLUT2.
Yes, only one gate, GLUT2.
Yes.
Okay, common exit pathway for all sugars, but entry is through different gates. Okay.
Yes.
Then which transporter is insulin dependent?
Option 2. Option B, GLUT2.
No. Insulin
GLUT1
dependent.
is for brain and RBC. GLUT2 is for pancreatic cells and liver cells. GLUT2, sir.
GLUT4. Okay, GLUT4 is present in the muscle and adipose tissue. Okay. Next.
Net ATP produced from glycolysis under anaerobic conditions is 2. Option B.
Correct, correct. So under anaerobic condition, you get only 2 ATPs. Total are 4 produced, but 2 are consumed. Okay.
Yes.
So net
in absence of oxygen, pyruvate is converted to lactate. Option C.
Correct. So that regenerates NAD plus, allows glycolysis to continue. Next.
ATP yield from complete oxidation of one glucose molecule is option C, 32 ATP.
Correct. As per the recent calculation, it is 32 ATP. Okay. 32 ATP. Modern accepted value is 32. Includes glycolysis plus TCA plus electron transport chain. Okay. So that finishes the glycolysis. Now we will begin with next topic, that is citric acid cycle, also called as Krebs cycle. Now, what is the location of this Krebs cycle? It happens into the mitochondrial matrix. Okay. So it happens into the mitochondrial matrix. So function is a complete oxidation of the acetyl CoA with production of electron carriers. So citric acid cycle, we have seen up till now what is produced. Glucose is producing two molecules of pyruvate. This pyruvate can undergo either aerobic condition, further process, or anaerobic condition. Now, when there is oxygen which is available, it can undergo this citric acid cycle or the Krebs cycle. So, where is the location of this event which is happening? It is happening in the mitochondrial matrix, and function is the complete oxidation of the acetyl CoA, then production of the electron carriers. Now, what are the products which are formed? You get a small amount of ATP, you get NADH and FADH2. Okay, so this also will give you energy, okay, in form of ATP, because one NADH is equal to 2.5 ATPs, one FADH2 equal to 1.5 ATPs. Now let us see this TCA cycle, also called as citric acid cycle, also called as Krebs cycle. Okay, so this is the Krebs cycle. So this is the pyruvic acid which is getting formed in the glycolysis process. So this pyruvic acid or the pyruvate is getting converted into the acetyl CoA. Okay, and here one NADH is formed. Okay, then this acetyl CoA combines with oxaloacetic acid and forms the citric acid, and your cycle begins. Okay, so what happens in this cycle? Then there are successive steps. Okay, total there are 8 steps in this cycle, out of which you see this particular step here, you get NADH and you get CO2 formed. Here also you get one NADH and one CO2 which is getting formed. Okay, in this step you are getting one FADH2. Okay, and here also you are getting one NADH. So in this cycle you are getting NADH 1, 2 and 3 places. So in the cycle you are getting NADH at 3 places, FADH at 1 place. Remember these particular steps. Okay, so the same thing we will see now in this particular format. Okay, so this is the acetyl CoA which is entering into this citric acid cycle, and this further goes through this cycle. Okay, and in this cycle you have one NAD which is getting formed here, and another also CO2 which is getting formed here, one NADH getting formed here, CO2 which is getting formed here, and here is one FADH2 which is getting formed. Okay, so acetyl CoA, that is 2 carbon compound, enters this cycle, and there are release of CO2 in step 3 and step 4. So step 3 and step 4 will release carbon dioxide. So here carbon dioxide is getting released, and here also carbon dioxide is getting released. Okay, now acetyl CoA is completely oxidized by this cycle and reaches alpha ketoglutarate stage. So you have got this acetyl CoA finally getting converted into alpha ketoglutarate. Okay, so ultimately you get this alpha ketoglutarate. Just hold on. And all reactions are reversible except step 1 and 4 step 1 and step 4. So here all stages are reversible except the step 1 and the step 4 are irreversible steps. Okay, so that is what is the Krebs cycle. Now what is the significance of the citric acid cycle? It leads to the complete oxidation of acetyl CoA. it forms the ATP. Then it is the final common oxidative pathway. It is the integration of the major metabolic pathways. Fat is burned on the wick of the carbohydrates here. Excess carbohydrates are converted as a neutral fat. No net synthesis of carbohydrates from the fat. Carbon skeletons of amino acid finally enter the citric acid cycle. It is an amphibolic pathway and it is anaphyliotic role. Okay. Now let us see the ATP generation steps. Okay, now concentrate on the step 3 and step 4. Okay, then step 3 and step 4, you have got 1,1 NADH which is getting formed. And as per the new recent 1 NADH is equal to 2.5 ATP. So step 3 is giving 2.5 ATP. Step 4 is also giving 2.5 ATPs. So step 3 is giving 2.5 ATPs and step 4 is also giving 2.5 ATPs. Okay. Then there is a 1 GTP which is getting formed. 1 GTP is equal to 1 ATP. So 1 ATP is formed here. Okay. So there is a 1 GTP. Okay. 1 GTP is getting formed in the step 5. Okay. That is 1 ATP. Then you have 1 FADH2 in step 6. Here also you have 1 FADH2 here. Okay. In step 6, that is equal to 1.5 ATP and total you are getting 2. Okay. Then malate final step is also giving 1 NADH. So this is the final step that is also giving 1 NADH. So that is equal to 2.5 ATP. Okay. So 1 NADH is equal to 2.5 ATP. Okay. So you have got how many ATPs total in the Krebs cycle? 10 ATPs as per the new calculation and 12 ATPs as per the old calculation. Okay. So total number of ATPs produced in the Krebs cycle are 10. Then the questions about the Krebs cycle. Doctor, can you please read the questions about the Krebs cycle? Next metabolic pathway for glucose is a Cori cycle or it is a lactic acid cycle. Okay. Cori cycle or lactic acid cycle. So this cycle is between the muscle and the liver. Okay. So in the muscle, glucose is getting converted into lactate. Okay. Now this lactate will go to the liver and from lactate you will get further glucose via gluconeogenesis. So this is what is the flow, what happens in the Cori cycle. So you in the muscle, the glucose is getting converted into the pyruvate via glycolysis. Again, this pyruvate is converted into lactate in the muscle under the anaerobic condition. Now, this lactate via the blood is transported to the liver. The liver will convert lactate again into pyruvate, and this pyruvate again in the liver will get converted into glucose via gluconeogenesis. So here is a new process of gluconeogenesis. There is a difference between the glycogenolysis and the gluconeogenesis. This gluconeogenesis is from non-carbohydrate source. Okay, maybe from acetyl CoA or maybe from amino acids. When the glucose is getting formed either from fat source or the fat source or the protein source, in that particular case we'll call this as a gluconeogenesis. If it is getting from carbohydrate source, that is mainly glycogen store, that is called as glycogenolysis. Okay, so this now pyruvate is getting converted into glucose. This will go via blood to the muscle, and again muscle will use the glucose. So we started in the muscle with the glucose and we entered back with the glucose reaching to the muscle again. So this is the Cori cycle. Okay. So it occurs during anaerobic conditions like vigorous exercise. It prevents the lactate accumulation in the muscle and glycolysis in the muscle and the gluconeogenesis in the liver. They are linked to each other. Okay. So these two processes, the glycolysis of the muscle and gluconeogenesis of the liver, they are interlinked via this Cori's cycle. Okay. So site of gluconeogenesis is the liver. Product leaving the muscle is the lactate. Energy status is, it is an energy consuming cycle. So this cycle is not, Cori cycle is not producing any energy, but it is energy consuming the cycle. Cause of oxygen debt is lactate metabolism and trigger is anaerobic exercise. So muscle makes the lactate, liver clears this lactate. It occurs in the anaerobic glycolysis. Cori cycle is equal to glucose lactate shuttle. Okay, so it is a glucose lactate shuttle which is running. Okay, so it causes the ATP. That is a burden to the liver and it is responsible for oxygen debt. Now we will discuss about the lactic acidosis and the glycolytic enzymes disorder. Okay, so there are various enzymes. If they are deficient in the glycolytic pathway, it can lead to the lactic acidosis. So first is a lactic acidosis. What is the definition? It is the accumulation of lactate in the blood. It will lead to the decrease in the pH, that is metabolic acidosis. And this is seen in various conditions such as hypoxia, shock, pulmonary failure, alcohol abuse, diabetes mellitus, etc. And the commonest cause is the hypoxia. Okay. Once there is a decrease in the oxygen, there will be increase anaerobic glycolysis. So more amount of pyruvate is getting formed. So this pyruvate will get converted into lactate via lactate dehydrogenase. Then this lactate will accumulate and it will lead to acidosis. So any condition causing tissue hypoxia, you think of lactic acidosis. Okay. So next is a glycolytic enzyme deficiencies. The general concept is that RBCs will depend on the glycolysis only. There are no mitochondria. So other Krebs cycle will not happen there. Okay. So only glycolysis will happen. So the ATPs are less amount is reduced. So RBC will be destroyed and it will lead to the hemolytic anemia. So what are the enzymes which are deficient and what will be the clinical picture? So we have seen PK, HK, PFK. These were the enzymes of the glycolysis. So there can be deficiency of pyruvate kinase that will lead to the decrease in production of the ATP and it reduces the affinity. So affinity towards the oxygen affinity will be reduced and there will be shifting of the oxygen dissociation curve to the right, but it will lead to the hemolytic anemia. While the hexokinase, that will also reduce the glycolysis entry and that will lead to the decrease in 2,3-BPG level, but it will shift the oxygen dissociation curve to the left. Okay, there is an increased affinity for the oxygen, but still there is a hemolytic anemia. So pyruvate kinase will reduce the affinity for the oxygen, while hexokinase will increase the affinity for the oxygen. With pyruvate kinase deficiency, there is a shift to the right for the oxygen dissociation curve. For the hexokinase deficiency, there is a shift to the left for the oxygen dissociation curve. But clinically, both will manifest as a hemolytic anemia. Then there can be deficiency of aldolase, that is impaired glycolysis, and you will again get the hemolysis here. Next is a PFK, that is the phosphofructokinase, that is the rate limiting enzyme, and this is the rate limiting step. If it is blocked, then you get muscle weakness. Okay, so these are the key enzyme and their deficiency. Okay, now this same thing what we have seen here. The first is a pyruvate kinase deficiency. Most common glycolic defect is pyruvate kinase deficiency. That will increase 2,3 BPG level, will decrease the hemoglobin affinity for the oxygen. There will be better tissue oxygen delivery. However, still there will be hemolysis because still it causes hemolysis due to ATP depletion. Next is a hexokinase deficiency. It will increase the affinity for the oxygen. So HB affinity for oxygen is increased. So there will be impaired delivery of the oxygen to the tissue. And next is a PFK deficiency. This is also called as Tori's disease. It affects the muscle and there will be severe exercise intolerance and the muscle weakness. Next deficiency is a pyruvate dehydrogenase, that is PDH deficiency. It converts pyruvate to acetyl-CoA. That is the link between the glycolysis and TCA cycle is via PDH, this enzyme. Okay, so this enzyme where the cofactor is important, that is a thiamine pyrophosphate that we have seen. Okay, so clinically, if there is a thiamine deficiency, which is seen most commonly in chronic alcoholism, malnutrition, that will lead to the decreased PDH activity and increased pyruvate level, so increased lactic acid level and leading to lactic acidosis. This is associated with beriberi. So PDH deficiency, pyruvate cannot enter into the TCA cycle, so it is shunted to lactate and it leads to the lactic acidosis. Okay, so alcoholic confusion, lactic acidosis, think of thiamine deficiency. PDH deficiency, neurological defects plus lactic acidosis. And TPP is equal to essential cofactor for PDH. Okay, this is what we have seen. TPP is a cofactor for this thiamine pyrophosphate. TPP is an important cofactor for PDH. Okay, so lactic acidosis is seen in hypoxia, shock, alcoholism. RBC energy failure will lead to hemolysis. PK deficiency, pyruvate kinase deficiency will lead to decreased oxygen affinity and increased levels of 2,3-BPG, while hexokinase deficiency will lead to increased oxygen affinity, okay, and decrease in 2,3-BPG level, but both will cause the hemolysis. PFK deficiency will cause muscle weakness. PDH needs thiamine. Alcoholism will have TPP deficiency leading to lactic acidosis. Okay, so we finished here most of the theoretical part. Doctor, you have few questions. Please go through these questions.
A 32-year-old woman is admitted with septic shock. Blood test shows metabolic acidosis and elevated lactate level. What is the most likely mechanism for these findings? Increase oxidative phosphorylation, conversion of lactate to pyruvate in liver, increase anaerobic glycolysis due to hypoxia, increase activity of pyruvate dehydrogenase, decrease glucose uptake by tissues. Option C, increase anaerobic glycolysis due to hypoxia.
Yeah, that we have seen hypoxia is the commonest. Increase anaerobic glycolysis. Here, anaerobic glycolysis is there due to hypoxia. Okay, so ultimately, yeah, it is a hypoxia which is there. So that leads to the increase anaerobic glycolysis. So option C, increase anaerobic glycolysis due to hypoxia. In shock, there is a tissue hypoxia, cells switch to anaerobic glycolysis, and pyruvate is converted to lactate and leading to lactic acidosis. This is the most common mechanism. Okay, so hypoxia will lead to lactate accumulation. Next.
A patient presents with chronic hemolytic anemia. Laboratory finding shows increased level of 2,3 BPG in red blood cells. Which enzyme deficiency is most likely? Phosphofructokinase. Option B, pyruvate kinase.
B.
B.
Right, your answer is B. So pyruvate kinase deficiency. Pyruvate kinase deficiency will lead to decrease in ATP in RBCs leading to hemolysis, and you have increased levels of 2,3-BPG, and there is a decrease in oxygen affinity. So you get a right shift of the oxygen dissociation curve. So increase 2,3-BPG plus hemolysis, pyruvate kinase deficiency. Next.
A chronic alcoholic patient presents with confusion and lactic acidosis. Which of the following enzyme activities is most likely reduced? Option A, hexokinase, lactate dehydrogenase, pyruvate dehydrogenase, phosphofructokinase, and option E, aldolase. Confusion and lactic acidosis. Thiamine deficiency.
Yes.
Option D, phosphofructokinase.
Is it phosphofructokinase or
Pyruvate
PDH? Okay.
PDH, yes.
PDH. Okay. We have seen for PDH. So for PDH, TPP is there, no? Thiamine pyrophosphate. So alcoholism lead to thiamine deficiency. Thiamine pyrophosphate is a cofactor for pyruvate dehydrogenase. So decreased PDH will lead to pyruvate accumulation, which is getting converted to lactate, leading to lactic acidosis. So alcoholic plus lactic acidosis, think PDH inhibition. Okay. Now we will start with next topic, that is electron transport chain. Okay, so what is electron transport chain? It is the final step of energy production. It converts energy from NADH and FADH to ATP. Okay, so if you want to understand this, ETC uses oxygen to generate maximum ATP. Okay, so what is getting utilized is the oxygen to generate maximum number of ATPs. And what is the location of this? It is the inner mitochondrial membrane. Okay, so this is the outer mitochondrial membrane, and this is the inner mitochondrial membrane. So this is electron transport chain is located here across the inner mitochondrial membrane. Okay, so where does it happen? It happens in the inner mitochondrial membrane. NADH and FADH2 will donate the electrons. These electrons will pass through the carriers, and final electron acceptor is the oxygen. The final process is the oxidative phosphorylation. So finally what happens is the oxidative phosphorylation. Final electron acceptor is the oxygen. Okay, so NADH2 and NADH and FADH2, they will donate the electron. We have seen NADH, 3 NADH are getting formed, 1 FADH2 is getting formed in the Krebs cycle. Okay, so let us look at this particular figure. Okay, now what you have, this is the inner mitochondrial membrane. This is the matrix. So this side is the matrix of the mitochondria, and this is the outer mitochondrial membrane, and this is the in between the outer and the inner membrane, there is a mitochondrial space. Okay, now here the this process of electron transport chain is located on the inner mitochondrial membrane, and basically there are 4 complexes on this inner mitochondrial membrane. See, here is a complex 1, then there is a complex 2, then there is a complex 3 and complex 4. So here is a complex 3 and here is a complex 4. Complex 1, 2, 3 and 4. These are the 4 complexes across the inner mitochondrial membrane. Now, what is the role of complex 1? Here, it will accept the NAD, okay, NADH. Okay, so NADH will come through this gate, then FADH will come from this particular gate. Okay, so what is happening? This NADH will enter through complex 1. So NADH will give the electron to the complex 1. So NADH will become now NAD plus. So NADH drops electrons at complex 1. Okay, so this NADH will drop 1 electron at complex 1 and will form the NAD plus. This is the first step. Okay, and whenever there is a electron which is getting donated, at that time there is a pumping of the proton from inside to the outside. There is a 4 protons which are getting pumped out when there is a transfer of electron from NADH to NAD, NADH to complex 1. Okay, so here NADH will give 1 electron to the complex 1. NADH will become NAD plus, and NADH drops electrons at complex 1. This is what is happening. Okay, then for complex 2 you have FADH2. So FADH2 comes from the Krebs cycle. Okay, it enters at complex 2. Okay, complex 2 does not pump the protons. This is the only site where the protons are not pumped even if the electron is getting donated. Okay, now at all complexes the protons are pumped out. Okay, at complex 1 the 4 protons are pumped out. Here in 3 complex, 4 protons are pumped out. At complex 4, 2 protons are pumped out, but at complex 2, no protons are pumped out and electron is donated by the FADH2. Okay, so this is what is happening at complex 2. Now, once at NADH is entering at complex 1, FADH2 is giving electrons at complex 2. Now, these electrons will be transported via CoQ shuttle. So there is a shuttle for the transport of these electrons to complex 3. So this will happen via CoQ shuttle. Okay, so this CoQ acts as a shuttle to transport these electrons to the complex 3. Okay, so what happens further is the electron flow, shows the path. Complex 1 and complex 2 via CoQ will give the electrons to the complex 3, and from complex 3 via cytochrome C they will go to the complex 4, and finally it will go to the oxygen. So final electron acceptor is the oxygen. But before this there is a complex 1 where the NADH gives the electron, complex 2 where the FADH2 gives the electron. At complex 1 4 protons are pumped out, at complex 2 no protons are pumped out. Then the CoQ shuttle will transport these electrons of complex 1 and complex 2 to complex 3. Then via cytochrome C they will go to the complex 4, again here 2 electrons will be pumped out, and final electrons will be accepted at by the oxygen. Okay, so at 1 you have 4 protons getting pumped out, at 3 you have 4 protons getting pumped out, and at complex 4 2 protons are getting pumped out. Okay, so coenzyme Q carries electrons from 1 and 2 to 3. Cytochrome carries the electron from complex 3 to complex 4. These are like shuttle buses carrying the electrons. Okay, now next step is a proton pumping. So this is the most important concept. Okay, where protons are pumped, complex 1 4 protons are pumped out, complex 3 4 protons are pumped out, and at complex 4 2 protons are pumped out. At complex 2 no pumping of the protons is done. Okay, so as the electrons move, the protons are getting pushed up, so it creates an energy gradient. Okay, so final step is oxygen accept the electrons and it will form the water. Oxygen is the final electron acceptor. Without it, everything will stop. So oxygen is a must. So this will happen in the aerobic conditions only. Now it is linked to the ATP. Okay, actually this is not shown in the diagram, but finally it is linked to the ATP. The explanation is that proton accumulation will create a gradient. This will drive the ATP formation via the ATP synthesis. So finally the ATPs are getting formed. So the protons want to come back. That energy makes ATP. So there was a gradient which was created because the protons were driven out. They want to come back, and that will create the ATP. Okay, so let us summarize. NADH enters at complex 1, FADH2 at complex 2. Electrons move via CoQ to the complex 3 from 1 and 2. Okay, and from 3 via cytochrome C, it will go to the complex 4. Protons are pumped out at 1, 3, and 4 complexes. Oxygen is the final acceptor. It forms the water, and there is a proton gradient, and this proton gradient will help in the ATP production. This is about the electron transport chain. Okay, so one thing you should remember, complex 2 does not pump the protons. Oxygen is required at complex 4. CoQ and cytochrome C, they are the mobile carriers for the electron. Then it is happening at the inner mitochondrial membrane. Okay, so the electron flow is push protons up. When they flow back, it will generate the ATP. Okay, from exam point of view, you should remember carbohydrates consist of carbon, hydrogen, and oxygen atom. Glucose is the primary energy source for the body. Digestion begins in the mouth through the salivary amylase. Major energy pathway is a glycolysis, then it is a TCA cycle, and then it is the electron transport chain. And we have seen this in this sequence only. There are other minor pathways also that we'll see later on. Okay, so maximum ATP is produced in oxidative phosphorylation. GLUT2 is present in the intestine, liver, and pancreas. It works as a high capacity transporter. Okay, this also we have seen in the beginning, that is the carbohydrate digestion in 3 sentences. The starch will get converted into disaccharides through the action of the amylase. Disaccharides will get converted into monosaccharides in the brush border enzymes. Then monosaccharides will get absorbed via the jejunum via the transporters. Which are the transporters? That also we have seen. So this flow chart will show the dietary carbohydrates getting converted, that is the starch, sucrose, and lactose in the mouth via the action of the salivary amylase or the ptyalin. They will getting formed into the maltose and the dextrins. So in the stomach, HCl, that is hydrochloric acid, will inactivate the salivary amylase. There is no significant carbohydrate digestion in the stomach. Then in the small intestine and duodenum, pancreatic amylases are there. They will convert the starch into maltose plus oligosaccharides. And in the brush border enzymes, or in the jejunum, there is a maltase which will convert maltose into 2 molecules of glucose. There is a sucrase which will convert sucrose into glucose plus fructose, and there is a lactase which will convert lactose into glucose plus galactose. And finally, monosaccharides will be formed, which are glucose, galactose, and fructose, and these will get absorbed in the intestinal cells. Okay, here also there are transporters which we have seen. Glucose plus galactose will go via SGLT1, that is a sodium dependent active transport. The fructose will go via GLUT5, that is a facilitated diffusion, and it will exit to the blood. While GLUT2 transporter, via the GLUT2 transporter, it will go into the portal circulation and then it will go to the liver. Okay. A small topic is a glucose alanine cycle, that is a Cahill cycle. Okay. So pyruvate is getting converted in the to alanine in the muscle. This alanine goes to the liver and again getting converted into pyruvate, and from pyruvate to glucose, that is the Cahill cycle. So importance is that nitrogen, there is a transfer of the nitrogen in the liver and glucose goes to the muscle. Okay. Now let us see the substrates in detail. So lactate from the anaerobic glycolysis is converted to pyruvate, and then from pyruvate you get glucose. While amino acid alanine is the major substrate here. Okay, it is seen in, so amino acids alanine is the major substrate. So we have finished this minor pathways also. We have few questions, doctor. Will you please start the questions, doctor?
Yes, sir. A 25-year-old woman is undergoing prolonged fasting. Gluconeogenesis is activated to maintain blood glucose level. Which of the following enzyme enables the liver to release free glucose in the bloodstream? Hexokinase, phosphofructokinase, glucose 6-phosphatase, pyruvate kinase, or option E, aldolase, glucose 6-phosphatase.
Because we are dealing with the gluconeogenesis. Okay. In gluconeogenesis, glucose 6-phosphatase is the enzyme. Okay.
Yes.
So converts glucose 6-phosphate to glucose. So it is present in the liver, but it is not present in the muscle. So it allows glucose release into the blood. Okay. So only liver can maintain the blood glucose due to glucose 6-phosphatase. Next question, doctor.
A patient with prolonged starvation is relying on gluconeogenesis. Which of the following substrate contributes most to glucose production from muscle? Most to glucose production from muscle. Lactate, option B, alanine.
Which is?
Option
Which
B,
is
sir.
the A or B?
Option B, sir.
Yes, alanine. Okay. So alanine is the major gluconeogenic substrate from the muscle. It is involved in the glucose alanine or Cahill cycle, transfers nitrogen from the muscle to the liver, and muscle to alanine goes to the liver and it produces the glucose. Next.
A researcher studies regulation of gluconeogenesis. Which of the following factor inhibits gluconeogenesis by acting on fructose 1,6-bisphosphatase? Which of the following factor inhibits gluconeogenesis by acting on fructose 1,6-bisphosphatase? Option B. Option. Which of the following factors inhibits gluconeogenesis by acting on fructose 1,6-bisphosphatase? Option D, AMP.
Yeah, cyclic, it is AMP. Okay. Fructose 1,6-bisphosphatase is inhibited by AMP and fructose 2,6-bisphosphate. So it is activated by citrate. Okay. So AMP is low energy. It will stop the gluconeogenesis. So glucose 6-phosphatase is present only in the liver. Alanine is major muscle substrate. AMP inhibits the gluconeogenesis. This is the take-home message. Okay. Now the last small topic is a glycogenesis, that is the synthesis of the glucose. Okay. The core concept here is that it is a formation of glycogen from the glucose. It occurs in the liver and the muscle. Steps of the glycogenesis are: first is the activation of the glucose. So glucose is converted into UDP glucose. So glucose 1-phosphate will combine with UTP and you will get UDP glucose. That is the activation of the glucose. The enzyme responsible is UDP glucose pyrophosphorylase. So UDP glucose pyrophosphorylase is the enzyme which will form the UDP glucose. So UDP glucose is the activated form of glucose. Now is the next step. That is the glycogen primer formation. Next step is a glycogen primer formation. This glycogen primer is nothing but the glycogenin. Okay, glycogenin. So it adds initial 7 glucose residues. So glycogen synthesis cannot start without this primer, that is glycogenin. That is the primer is getting formed. First was the activation of the glucose and formation of the UDP glucose. Then it will add 7 glucose residues and form the primer, that is glycogenin. This glycogen synthesis will, after this it will start. Okay. So next is a chain elongation. Now you have got a basic workup done. After that, the chain will get elongated. The enzyme responsible is a glycogen synthesis, and it will form the alpha 1,4 glycosidic bonds, and it adds glucose to the non-reducing ends. Okay. So rate limiting enzyme here is a glycogen synthase. Okay. It is the elongation of the chain. Only glucose molecules will get added, and the enzyme is a glycogen synthase. Next is a branch formation. So enzyme is a branching enzyme. You will get amylo 1,4 to 1,6 transglycosidase, and it creates alpha 1,6 linkages, and it transfers 6 to 8 glucose units. Okay. So branching will lead to increase in solubility and rapid mobilization. So activated substrate is a UDP glucose. Primer required is a glycogenin. The main enzyme is the glycogen synthase. Bond which is formed is a alpha 1,4 bond. Branching enzyme is a alpha 1,6 linkage, and site is a liver and muscle. Okay. Last concept is a glycogenolysis. We have just seen that the glycogen is the storage form. How it gets formed, that we have seen. Now, how this glycogen is broken down. The core concept is a breakdown of the glycogen to glucose 1-phosphate. It maintains the blood glucose, and this happens mainly in the liver, and it will provide the energy to the muscle. Okay. So key steps are glycogen phosphorylase. That is the main enzyme. It cleaves the alpha 1,4 bonds and produces glucose 1-phosphate. So the rate limiting enzyme is a glycogen phosphorylase. So for glycogenolysis, the enzyme is a glycogen phosphorylase. Then comes the debranching enzyme. It removes the alpha 1,6 branches, and it is required to completely degrade the glycogen. Next is a conversion to glucose. Glucose 1-phosphate is converted to glucose 6-phosphate. In liver, glucose 6-phosphatase is present that will free the glucose from the glucose 6-phosphatase. So glucose 6-phosphatase. So muscle lacks the glucose 6-phosphatase, that also we have seen. Now, how this is regulated? There is a reciprocal regulation. Okay. Now, how this reciprocal or the covalent regulation is there? Let us see. You have glycogen phosphorylase that will, after phosphorylation, that will active when phosphorylated. So this enzyme, when phosphorylated, it will be active, glycogen phosphorylase. Okay. So it will lead to glycogenolysis. Glycogen phosphorylase will get phosphorylated and will lead to glycogenolysis. While the glycogen synthase, when it gets phosphorylated, it will become inactive when it gets phosphorylated. So it will stop the glycogen synthesis. Okay. That is how you maintain the level. Now there is a hormonal control also. Glucagon will increase glycogenolysis. Adrenaline will also increase the glycogenolysis, while insulin will lead to increase in glycogenesis. So it will increase the carbohydrate metabolism store. Okay. It is mediated via cyclic AMP. Now there is a allosteric regulation also. Allosteric regulation means by acting or by joining at another site apart from routine site. Okay. So that is glycogen phosphorylase will be inhibited by ATP, activated by AMP, and inhibited by glucose 6-phosphate. Okay. And even calcium is also activating glycogen phosphorylase, while glycogen synthase is inhibited by ATP and AMP and activated by glucose 6-phosphate and the calcium. So this glycogenesis and glycogenolysis that we have just seen, It is the storage of glucose in form of glycogenesis. So glycogenesis is the storage form of glucose, and key enzyme is the glycogen synthase, and hormone is insulin hormone, which is responsible. Phosphorylation leads to the inactivation of the hormone, and product is the glycogen. While glycogenolysis is the breakdown of the glycogen, the main enzyme is the glycogen phosphorylase, and glucagon and adrenaline will lead to increased activity of this enzyme, and phosphorylation, the activity of this enzyme will increase, and product is glucose 1-phosphate. Okay, so just recapitulate the things. UDP glucose is the activated form of glucose. Glycogen synthase will lead to the synthesis of the glycogen. Then phosphorylase will lead to the breakdown, that is glycogenolysis. Phosphorylation on lead to the glycogenolysis. Phosphorylation off leads to the glycogenesis. Okay, liver has glucose 6-phosphatase, but muscle doesn't have. Doctor, can you read these questions?
Yes, sir. 28-year-old woman in the fed state is storing glucose as glycogen. Which enzyme is primarily responsible for elongation of glycogen chain? Glycogen phosphorylase, glycogen synthase, hexokinase, glucose 6-phosphatase, or phosphofructokinase 1. So option B, glycogen synthase.
Correct. First thing, concentrate that which state we are talking about. Then what will be the event which is involved? It is a fed state. So glucose will get converted into glycogen, and enzyme they are asking for elongation. It is a glycogen synthase. Okay. So it adds glucose units via alpha 1,4 bonds. That is the rate limiting enzyme for glycogenesis. Next.
During glycogen synthesis, glucose must first be activated, which is the activated form used for glycogen formation. Sir, option B, glycogen activated state, UDP glucose. Option C, UDP glucose, sir.
Yes, UDP glucose, that is the first step. Okay, UDP glucose is the activated glucose donor formed using UTP. Next.
A defect in the branching enzyme will most directly affect which of the following linkages? Sir, option C, alpha 1,6 glycosidic bond.
Is it A or C?
Sir,
branching enzyme.
Branching option C only, 1,6.
Option 6. Okay, alpha 1,6 glycosidic bond. So branching enzyme forms alpha 1,6 bonds, important for the branch formation. Next.
Which of the following best describes the effect of phosphorylation of glycogen metabolism enzyme? Phosphorylation of phosphorylation on glycogen metabolism enzyme. Activates glycogen synthase, inhibits glycogen phosphorylase, activates glycogen phosphorylase, activates both enzyme, inhibits both enzyme. So option C, activates glycogen phosphorylase.
Correct. So it will activate the glycogen phosphorylase. Okay, so phosphorylation will activate glycogen breakdown. So glycogen synthase will become inactive, and other hormone will become phosphorylase will be active. Okay, next.
A muscle cell is actively breaking down glycogen during exercise. Why can it not release free glucose into the bloodstream? A muscle cell is actively breaking down glycogen during exercise. Yes, why can it not release free glucose in the bloodstream? Lack of glycogen phosphorylase, lack of UDP glucose, absence of glucose 6-phosphatase, absence of ATP. Sir, option C, absence of glucose 6-phosphatase.
Yes, absence of glucose 6-phosphatase. So muscle cannot convert glucose 6-phosphate to glucose, hence cannot maintain blood glucose. Next option.
Which of the following molecules activates glycogen phosphorylase in muscle? ATP, glucose 6-phosphate, AMP, insulin citrate. Which of the following molecule activates glycogen phosphorylase? ATP, sir, option A.
Which of the following molecule activates glycogen
Glycogen.
phosphorylase in muscle? It's AMP, doctor. Low energy. AMP is low energy. It will stimulate the glycogen breakdown. Okay.
Yes, sir.
Next.
A patient with hypoglycemia has increased glucagon secretion. With what is the primary effect on glycogen metabolism in the liver? Increased glycogenesis, decreased glycogenolysis, increased glycogenolysis, inhibition of CMP production, activation of glycogen synthase. Sir, increased glycogenolysis.
C, because it is a glucagon. Okay, so
Glucagon,
glycogenolysis will increase. Glucagon will increase cyclic AMP. Phosphorylation of the phosphorylation cascade will get activated, and it will activate the glycogen phosphorylase. Okay, so glycogen synthase will lead to synthesis of glycogen. Glycogen phosphorase will lead to breakdown of phosphorylase. Glycogen, the UDP glucose is the activated glucose. Alpha 1,6 bond lead to branch because of branching enzyme. AMP activates glycogenolysis. Muscle lacks glucose 6 phosphatase, and glucagon leads to glycogen breakdown. Okay, so that finishes the metabolism of carbohydrates. Thank you. There are various glycogen storage diseases. Okay, there are many. Actually, they are numbered from 1 to 8, 9, 10. But I am just going to discuss from the examination point of view, which are more frequently asked. You remember this 1, 2, 3, 5. Okay, 4 is not very common. Okay, that is why I have not included here 4. Okay, type 1 is a von Gerg's disease. Glucose 6 phosphatase is the enzyme which is deficient, and it causes severe hypoglycemia and hepatomegaly. Type 2 is a Pompe's disease that causes lysosomal deficiency of lysosomal maltase and leads to the cardiomyopathy. Okay, so remember Pompe's disease is type 2. There is a pump problem. Pump is a heart. That is why cardiomyopathy. Then type 3 is a Cori's disease. Okay, that is the debranching enzyme. Okay, and that causes hepatomegaly. Type 4, though I have not included, but that is a deficiency of a branching enzyme. So the first is a debranching is a third, branching is a fourth. And type 5 is a McArdle's disease, that is a muscle phosphorylase that leads to the exercise intolerance. Okay, so 1, 2, 5, even sometimes 3 they can ask. There is a mnemonic: very poor carbohydrate metabolism always. Okay, very poor carbohydrate metabolism always. V for von Gerg's disease, that is a type 1 glucose 6 phosphatase deficiency. Poor P, Pompe's, that is type 2, that is lysosomal alpha glucosidase deficiency and affecting the heart, causing cardiomyopathy. C is a Cori's, that is type 3 disease, debranching enzyme. And fourth I have not included. Fifth is a McArdle, that is a type 5, that is a muscle phosphorylase enzyme deficiency. And the type 4, which I said is not very common, is A. Okay, always metabolism always. So always A is Anderson disease, that is type 4 disease, where the branching enzyme is the deficient enzyme. Okay, so you can remember this. Type 1 von Gerg's disease, Gerg glucose stuck. Glucose is stuck, that is why you get hypoglycemia, severe hypoglycemia leading to the lactic acidosis, and you get hepatomegaly. Then type 2 is a Pompe's disease. There is a pump problem, the heart. So you get a cardiomegaly, the muscle weakness, infantile form of fever, severe, infantile form is severe. Then type 3 is a Cori's disease, that is can't complete the breakdown. That will lead to the mild hypoglycemia and limit dextrin accumulation. Type 4 is not very common, but that is Anderson's disease or the abnormal structure, that is a branching enzyme is deficiency. In type 3, debranching. So debranching deficiency first, then the branching deficiency. Type 3 is a Cori's. Type 4 is Anderson. Okay, here defective branching enzyme, and there is a liver cirrhosis. Type 5 can also be asked, that is McArdle. Okay, McArdle is a muscle problem, exercise intolerance. So there is a muscle cramps and myoglobinuria. So if you remember this particular sentence as a mnemonic, so very poor carbohydrate metabolism always. Now this always is stands for type 4 and M stands for type 5. Okay, that is a McArdle's disease. Okay, so these are the 5 more frequently or the important from the examination point of view. That is why I have covered this. So glucose is stuck in the liver, type 1. Stored in the lysosome, affecting the heart, is type 2. Partially broken, that is the debranching enzyme, that is the type 3. Then poorly structured, that is the branching enzyme, type 4, and cannot be used in the muscle. Severe muscle weakness is in type 5, that is a McArdle's disease. Okay. So other way to remember is odd numbers, that is 1, 3, and 5. These are the cytoplasmic enzymes, and type 2 and type 4 enzyme deficiencies, they are structural or the lysosomal enzymes. Okay. Doctor, we have few SBS. Can you please read the SBS?
Yes, sir. The
Yeah.
electron transport chain is located in which part of the cell? In a mitochondrial membrane.
Yes, option C, inner mitochondrial membrane. I have already mentioned that all locations you should know. All ETC complexes 1 to 4 are embedded in the inner mitochondrial membrane, and they are critical for proton gradient formation. Next.
Which molecule acts as a final electron acceptor in the electron transport chain? Oxygen, option C.
Yeah. Oxygen accepts the electrons at the complex 4, forms water, and if it is absent, ETC will stop and there will not be production of ATP. Next.
Complex not pumping electrons. Which of the following complex does not contribute to proton pumping in ETC? Complex 2, option B.
Yeah, complex 2 transfers electrons but does not pump protons. Okay. Next.
ATP yield from one NADH molecule in ETC is 1.5 ATP.
How many?
2.5 ATP, sorry.
Correct.
Option D.
NADH is 2.5. Okay.
Yes.
This is what is the accepted, modern accepted. Initially, they were calculating as per 3. Okay. And that is why calculation was coming total 38. Now they are saying 1 NADH is equal to 2.5 ATP. So it is now 32 total
32.
ATPs. Next.
A child presents with severe fasting hypoglycemia, hepatomegaly, and lactic acidosis. The most likely enzyme deficiency is Gluconeogenesis disease. Option C, glucose 6 phosphatase.
Yeah, glucose 6 phosphatase. It is a von Ger's disease. Liver cannot release free glucose. There is a severe hypoglycemia associated with lactic acidosis. Next.
A patient develops muscle cramps and myoglobinuria after exercise. Blood glucose level is normal. The most likely diagnosis is McArdle's disease.
Correct. So answer is McArdle's disease. There is a muscle glycogen phosphorylase deficiency. Muscle cannot use glycogen, and exercise intolerance is there. There is no hypoglycemia. Okay, liver is not affected. Okay. So about the electron transport chain, you should remember the location, that is inner mitochondrial membrane. Final acceptor is oxygen. Complex 2 will not pump out the proton. NADH will give rise to 2.5 ATP. Okay. Type 1 is a von Ger's disease leading to hypoglycemia. Type 5 is a McArdle leading to muscle cramps. Okay.